and get started. So thank you everyone for joining us today for the Physiology Seminar Series, presented here by Dr. Gerald Shadel. Gerald earned his Ph.D. from Texas A&M right here in Texas. And then he completed his postdoc work with David Clayton, Dr. David Clayton at Stanford University. And Jerry joined the Department of Biochemistry at Emory as an assistant professor. And as an associate professor, he then moved to Yale. And he currently is at the Salk Institute of Biological Studies and holds the Audrey Giselle Chair in Biomedical Science. He's also the director of the San Diego Nathan Schock Center of Excellence in Basic Biology of Aging. Jerry has numerous awards. I won't go through the whole list, but some of them include the Award for Excellence in Teaching, Breakthrough in Gerontology, Amgen Outstanding Investigator Award, and so on. Jerry's work has used yeast, mammalian cells, and mouse models to study the mitochondrial gene expression and the role of mitochondria in disease pathology. And today, he will tell us more about the mitochondrial stress signaling in aging and immunity. And with that, it's all yours.
All right, thanks, Anju. Thanks for the invitation to speak today and tell you an update on some of our work on mitochondrial stress signaling. I wish I could be there in person to come back to Texas and get some barbecue and beer, but we'll do that next time, I guess. That's one thing I really miss about Texas is barbecue. Man, it was good there. All right, so yeah, my research program is really centered on the role of mitochondria these days in diseases, aging, and immunity. And mitochondria probably don't need much of an introduction, but I always start here to remind people that mitochondria are not little sausages in the cytoplasm. They're actually a dynamic network. They constantly fuse and divide and move around the cell, and their form dictates their function. I mean, so they change their shape and move around and do things in order to adjust to metabolic and stress signals and physiological signals. They're also quite tissue specific. You know, mitochondria aren't all the same in all cell types. You know, they may have a core set of proteins that are, make up sort of a standard mitochondria, but the tissue specificity of the proteome and morphology, et cetera, is quite remarkable, actually. They're quite diverse. And that makes sense because their metabolic activities and their signaling activities need to be tailored to each cell type. They're quite complicated. There's upwards of 1,200 proteins in a mitochondria typically. Most of those are encoded by genes in the nucleus and imported into one of the 4 compartments in the organelle. Certainly they're at the crossroads of metabolism. Most famous function, of course, is to make ATP through the process of oxidative phosphorylation or OXPHOS. But really there's dozens of other essential biochemical reactions that occur in mitochondria that are essential for cell physiology and impact disease and aging. Early 90s, Xiaodong Wang showed that one of the electron carriers in mitochondria actually can leave mitochondria and be a signal for the cell intrinsic pathways for apoptosis. So mitochondria are not only involved in cell life, but also in programmed cell death and many other types of cell death at this point. But where I've been really interested over the last 10 to 15 years is really to understand the role of mitochondria as a signaling entity, both as a receiver of signals from the cell and as a generator of signals that the cell listens to in order to respond to stress and other physiological parameters. And really, what's the role of these signaling pathways in diseases, immunity, and aging? But before I go on, I wanted to just give a little shout out to one of my heroes who actually was at UTSW. So many of you may, I'm sure, remember Ron Butow, who was at UTSW most of his career, if not all of it. And he really is the father of this idea that mitochondria can signal and defined this retrograde signaling idea. This is a paper in Science of his from 1987. The first year I was in graduate school. And I was really interested in this paper, even though I was working on bacteria at the time. I wasn't working on mitochondria. But if you read that last line there, it says yeast cells seem to respond to the quality and quantity of mitochondrial DNA and modulate the levels of nuclear encoded RNAs, perhaps as a mean of intergenomic regulation. So this was really a breakthrough paper suggesting that mitochondrial genome can communicate with the nuclear genome. And I had of course, once I got into David Clayton's lab, I started going to mitochondrial meetings. I got to meet Ron. I think I met him when I was a postdoc in probably 92, something like that. And he was over at the bar having a vodka and tonic or whatever he was drinking. So I went over there and I said, "Hi, I want to introduce myself. I'm Jerry Shadel. I'm just a big fan of your work and all this," a little groupie kind of thing. And I asked him, I said, "Hey, what made you start studying signaling in mitochondria?" And his answer was the following. He said, "Jerry, I always felt that mitochondria must do something else more interesting than just OXPHOS." He says, "Everybody focuses on OXPHOS." He said, they're complicated. They must do way more interesting things than just that. I thought that was the coolest answer. And so I ended up studying mitochondrial signaling. So he was a huge influence on me, and he was a great scientist. And I actually came to UTSW, I think in 2005 is the last time I've spoke here. He invited me, and we just had a really great time. I think it was a couple years before he passed away. But just wanted to dedicate this talk to him. All right, so what about this idea of signaling between the nucleus and mitochondria? So as Ron showed, changes in the mitochondrial genome can elicit changes in the nuclear genome way back in 1987. But of course, at this point, we know that there are signaling going both ways. The nucleus obviously is, most of the proteins in mitochondria are coming from the nucleus. And so if you really want to change, long-term, the structure, the composition of mitochondria, you have to change gene expression in the nucleus. That's what you have to do. So the idea is that stress signals from mitochondria signal back to the nucleus to enact those gene expression programs. And so there's a homeostatic back and forth here to maintain mitochondrial function or to change them in response to different physiological or pathological cues. And so at this point, we know there are many different types of signals that come from mitochondria that do this. One of the classic ones is activating AMP kinase, for example, in response to energy charge. We now know that there are peptides that are released from mitochondria. Metabolites can regulate transcription and epigenetic states in the nucleus. Reactive oxygen species, an area I work a lot in, can relay redox signals to the nucleus and change expression patterns. And what I'm going to talk about for the rest of the day, though, is the role of mitochondrial DNA itself as a retrograde signaling molecule and how we stumbled on that. So mitochondrial DNA in mammals is a 16.5 kilobase double-stranded circle. It encodes 37 genes, 13 mRNAs that are essential subunits of the OXPHOS complexes. Remember, there's about 80 of proteins in these complexes, so 13 come from the empty DNA. The rest come from the nucleus and assemble around those mitochondrial encoded subunits. So immediately, to make that system, you have to have intergenomic communication of some kind, right? To make proteins inside mitochondria, you need ribosomes. There's a completely unique set of ribosomes in mitochondria that are different from your cytoplasmic ribosomes, and they're different from bacteria too. In fact, there was a great paper on the cover of Science a couple weeks ago on the structure of the mitochondrial ribosome bound to the inner membrane, so you check that out. But to make a ribosome, of course, you need lots of proteins, and you need and there are 2 ribosomal RNAs encoded by the mitochondrial DNA. So there's really only 2 entities in your cells that have components from the nuclear and mitochondrial genome, the OXPHOS complexes and mitochondrial ribosomes. And of course, the last thing you need are tRNAs, and there's 22 tRNAs encoded by the mitochondrial genome. So mitochondrial DNA is another unique feature of it is that it's multi-copy, and I'm going to get into that per cell, so I'll get into that in a minute. But over here on the lower left is a DNA stain in an embryonic fibroblast. You can see the large nucleus there staining, but the speckles, these constellation of stars out in the cytoplasm are the mitochondrial DNA molecules packaged into what are called nucleoids, and that's going to be important. And Ron did a bunch of work on yeast nucleoids too, figuring out what the composition of those proteins that bind mitochondrial DNA are as well. All right, so mitochondrial DNA in mammals is maternally inherited. And as any DNA that's inherited, if it's mutated, can cause diseases. And so there are now at least 200 known pathogenic mutations in mitochondrial DNA that cause maternally inherited diseases. And these are complicated diseases. They're being unraveled by people like Ralph DeVerdinis here as we speak, but they're very complicated. They have complex genetics because of two reasons, really. The multi-copy nature, you can have mixtures of mutant and wild type, and that can drift, and all sorts of fun genetics there. So you can have these so-called heteroplasmic states that affect the expression of the disease pathology. They're also heavily influenced by the nuclear genetic background, as you can imagine, because most of the proteins, again, come from the nucleus, and so nuclear polymorphisms can affect mitochondrial diseases. They're heavily influenced by environmental factors, and oddly enough, they're quite tissue specific. A lot of the diseases, you would think if you had a mutation in your mitochondrial DNA, it would kind of just have a similar effect in all tissues, but it really doesn't. And I think the other important point I want to make is highlighted here is called the threshold effect. And that is with many mitochondrial mutations, mitochondrial DNA mutations, you have to reach a really high level of mutant genomes before you have disease pathology, which means that there's really quite a large buffer of OXPHOS activity that exists. And this plays into this multi-copy nature of the genome that I'm going to talk about in just a second. And of course, another important feature down the bottom here is that mitochondrial damage and mutagenesis increases significantly with age. And I think this is going to be important. I'll try to bring that point home at the end of the talk. All right, so what about this multi-copy genome? So most cells have thousands of copies of mitochondrial DNA. This is 100, so multiply by 10. And the mitochondrial genome is interesting in a couple of ways that are important for what I'm going to propose here. First, I've already said it's in excess of that needed to sustain oxidative phosphorylation. I'll give you another example of that in just a minute. It actually has fewer DNA repair pathways than the nuclear genome. So it does really nice base excision repair, but it's missing nucleotide excision repair, for example. So it's sort of replete or rather depleted in repair mechanisms. Finally, it has kind of a weird asymmetric DNA replication mechanism that leaves the genome single-stranded a lot during this process. And single-stranded DNA is more prone to damage than double-stranded DNA. So one question is, why have we maintained sort of a clunky, damage-prone cytoplasmic genome that is in vast excess of that needed for ATP production, which is what everybody says this genome does, and it does do that. So that's the question I want to pose. And my short answer, and I'll tell you the long answer in a minute, is that I think it's doing something else equally important that has been selected for. And so that's sort of the backdrop for the talk today. All right, so how do we get into this? So in my lab, we've been trying to make different cell and animal models of mitochondrial stress and ask, what are the signaling pathways that get enacted during that? How do nuclear gene expression change in response to different kinds of stress, et cetera? And the idea is if we can figure out those signal transduction pathways, those might be ones we can inhibit or activate in order to have beneficial effects on health. So one of the first models we made was a mouse model where we knocked out a nuclear gene called TFAM. So TFAM was discovered in David Clayton's lab right when I was a postdoc there, right before I was a postdoc there. And it's a DNA binding protein. It has 2 HMG DNA binding domains. It's a small protein, 25 kilodaltons, and it was originally identified as a transcriptional activator. It activates transcription of the promoters on mitochondrial DNA. So things encoded in the nucleus, goes in the matrix, binds to mitochondrial DNA, and activates transcription as part of the gene expression program. It also can repress transcription under certain circumstances, but the function of it that I want to highlight today is it has kind of a histone-like function, even though it's not a histone. It can bend and wrap the DNA and package. It's really the nucleating protein to package into these nucleoid structures that I referred to earlier. So if you knock TFAM out in a mouse completely, they die in embryogenesis. So you lose mitochondrial DNA, 0 oxphos, you're a dead mouse. So mitochondria are important, and oxphos is important. But what we found, and this is the work of Nils Larsen, who was a postdoc with me in David Clayton's lab at the same time. He's the one who made the knockout. He showed that the heterozygous animals really had no phenotype. But the surprising thing is they had half as much mitochondrial DNA as a wild-type mouse in most of their tissues, and even less than half in several tissues, up to like 30%. So I was always intrigued by that. It's like, how the heck why do we have thousands of copies of mitochondrial DNA per cell if you don't need half of them, at least to be a mouse in a cage? And so when I started my lab, I thought it would be cool to, or a few years after I started my lab, I thought it would be cool to ask, do cells from these mice even know they don't have enough mitochondrial DNA? And I thought also we'd sort of take a chapter from the Drosophila folks and say, hey, maybe this could be used as a sensitized background, a sensitized genetic background to identify pathways and processes where mitochondrial DNA instability might be important. So you might be able to uncover those more easily in such a background. So the first thing we actually did was cross the TFAM het animal, so missing one copy of the TFAM gene. And we crossed them to the APC-Min intestinal cancer model. And we published this, 10 years ago. And it wasn't a huge paper. There was a small effect, honestly. There was a slight increase in intestinal tumorigenesis in these mice, more prevalent in the distal part of the small intestine, and a little bit more in the colon, actually, which is a little bit unusual for this model. But the bottom line is, okay, proof of principle. In this, we unveiled a potential role for mitochondrial DNA instability in tumorigenesis. And so it was a really small paper, and we published that in the American Journal of Pathology, like I said, 10 years ago now. So about that time, a postdoc, Philip West, came into the lab, and he wanted to study mitochondrial stress pathways. And he says, why don't we just use the TFAM hets and just do gene expression profiling and look at the cells, what kind of, is there any stress? They don't have much of a phenotype. And so what he found is, so these are embryonic fibroblasts from TFAM mice, wild type and TFAM mice. And as you expect, you get half as much mitochondrial DNA. There's really not a major or significant effect on basal oxygen consumption. And he even showed that if you look at the transcripts coming off the mitochondrial genome, those 13 mRNAs, there's really no change in gene expression per se. So even though there was half as much DNA, the RNA output was basically the same. But what he did find when he looked at the cells, there was something going on. And you can tell in these wild type cells, now this is the picture I showed you a minute ago. If you look at the TFAM MEPs, you can see that these nucleoids, there's fewer of these nucleoids, and they appear larger. And the mitochondria go into a more hyperfused or stringy morphology. So he said, okay, they are experiencing stress. So he said, let's do gene expression profiling and see what's happening. And this turned out to be arguably the easiest gene expression pathway analysis you ever did, in that the top 45 or so upregulated genes in these cells were interferon stimulated genes. And so we're like scratching our head over what the heck is going on here? Why? And after I made sure that his cultures weren't contaminated with a virus or something, he assured me they weren't. And if you know Philip, they definitely weren't. And he confirmed all this with RT-PCR, and you do the pathway analysis, even though you didn't really have to. You get interferon signaling, you get pattern recognition receptor, innate immune activation. So it's as if these cells were infected like with a pathogen of some kind. And that would predict activation of interferon response factors in the STAT pathway, et cetera. So that's where we ended up, I should say. And it turns out that the role of mitochondria and innate immune signaling was already known. And that was really shown by here at UTSW with the discovery of MABs by James Chen, which MABs is an outer membrane protein. mitochondrial membrane protein, and it's an integrator of signals to the type 1 interferon and NF-kappa B pathway, downstream of, usually for MAV's case, a downstream of viral RNA entering a cell. So this is your innate immune system to RNA viruses. There's also a DNA sensing arm. Again, James Chen, a huge player in this, discovered cGAS, which is a major DNA sensor in the cytoplasm. And again, that's DNA in the cytoplasm gets sensed by cGAS, activates STING, and activates these same downstream pathways. So this was known. So our question was, we weren't infected, right? We were messing with the mitochondrial DNA with a TFAM deletion and getting activation of an interferon-like response. So the question was it this pathway or is it some novel pathway of activating the interferon response? It turns out it is this pathway. Philip knocked out IRF3, which is a major transcription factor that activates this response, and the response went away in the TFAM heterozygous MEFs. If you knock out TBK1, the kinase that phosphorylates IRF3, same thing. If you knock MAVs out, you get a slight diminution of the response, but it wasn't really fully responsible for it. But the cGAS STING knockouts kind of leveled this out. So it really looked like we were activating the cGAS STING pathway. So Philip went ahead and went with the hypothesis that somehow the mitochondrial DNA must be getting outside of mitochondria and binding to cGAS. And so what he showed with some really crucial and hard to do cytoplasmic fractionation experiments, highly purified cytoplasm was able to show that there was an increase in cytosolic mitochondrial DNA, both in MEFs and in bone marrow-derived macrophages, et cetera. So it looks like because of this perturbation in TFAM and this change in mitochondrial morphology, et cetera, that the DNA is gaining access to the cytoplasm and activating cGAS. As a consequence, we have sort of a basal increase in the interferon-stimulated gene response, and consequently, as you would predict, these cells are actually resistant to viruses. So if you infect them with any virus, even an RNA virus or a DNA virus, HSV or VSV here, these are GFP tagged viruses, you can see that the TFAM hets are quite resistant to viral infection in vitro. And even in vivo, if you infect them with LCMV virus, RNA virus, they're partially resistant to infection in vivo as well. Even more interesting is, now forget about the TFAM heterozygous situation for a moment. These are just wild type MEFs now, and we're infecting them with HSV, a DNA virus. And when you do that, look what happens. You get the TFAM phenotype. The nucleoids get larger, the mitochondria elongate, and TFAM's even depleted almost to the same degree as the TFAM het. So this is interesting, but if you do it with an RNA virus, in this case VSV, that does not happen. Okay, so somehow the TFAM hets look like they were mimicking to some degree a DNA viral infection because of the empty DNAs getting out. Furthermore, HSV actually encodes a protein called UL12.5 in the viral genome, and James Smiley actually showed that if he actually originally was the one to show if you infect with HSV for a long time, you actually completely lose the mitochondrial DNA. It gets degraded. And how that gets degraded is because of this UL12.5 protein. So he sent us some constructs where you just can express UL12.5 alone, and it phenocopies the TFAM perfectly. Increased size of the nucleoids initially goes away eventually. Elongated mitochondria and activation of the ISG response. So this is a really interesting, why a virus wants to do this is not clear to me, but there seems to be crosstalk and a battle going on between mitochondria and the viruses here. So our model is that, by some mechanism, depleting TFAM is leading to mitochondrial changes that allow the empty DNA to escape into the cytoplasm where it binds cGAS and activates and primes interferon response and an antiviral response, actually. So I just wanted to show you a movie. This is work by Laura Newman in my lab and in collaboration with Uri Manor. And we've been doing some live imaging to try to see if we can capture these release events and also some correlative light electron microscopy to try to figure out what exactly is going on. But here's, if you look at this nucleoid here, so this is, the DNA is stained in green. The mitochondria are stained with TOM20, which is an outer membrane marker, so that's important. So you can see the limits of the mitochondria, and you'll see this nucleoid escape from the mitochondria and then become free in the cytoplasm. So now it's out and it's moving around. So we know when that happens that C-gas is on it pretty quickly, and that the signaling is beginning. But we're still unraveling precisely what the release mechanism is in this circumstance. All right, so I was asked to provide a pause point for any questions. I don't know if there's any questions before I move on, because there's sort of a second half into this story. This might be a good point to get any basic questions about the mtDNA or mtDNA release, if anybody has them. And
So Jerry, we have a question in the Q&A by Xing Zhang. And he asks, is DNA being specifically released from mitochondria of the TFAM het cells, or do other mitochondrial contents also get released?
Yeah, it's clear that we're not releasing cytochrome C and things of this nature, or we'd be inducing apoptosis. So I think this is in this particular case, is a mechanism for the DNA itself to be released and sensed. And we're wondering now if it's not some form of mitophagy, I guess micromitophagy, where we're trying to get rid of some damaged genomes. And so we're trying to figure out right now, what is the nature of those big nucleoids? You know, there's multiple ideas about how you can get these big nucleoids. Some of them are thought to be, they cluster together sometimes and get on top of each other under stress. But also after replication, the genomes, when you replicate a circle, you end up catenated. And you have to use topoisomerases to undo the genomes and put them back together and then segregate. And so I think maybe the TFAM, my guess is that the TFAM deficiency may be involved in that decatenation or in the segregation of the genomes as the mitochondria are trying to divide, which is based on some of the work Jody Nunari has done. And I think maybe they're elongated because they're at a checkpoint. They're waiting for the genomes to finish replicating and separate, and then they're going to divide and inherit a genome. But I think they're stuck in some sort of checkpoint there. So that's our hand-waving idea. We're looking for catenanes right now and trying to figure out exactly what the big nucleoids are. But those are going out, and TFAM's going with them. So those are the two things I know that are out there. And that makes sense because Veit Hornig showed that TFAM actually bends the DNA in a way that C-gas likes it. So I think there's a role for TFAM in how C-gas is recognizing that.
And we have a few more questions. Weibo Lu asks, how big are these DNA fragments? And how are these mitochondrial DNA cleaved in the TFAM het cells?
Yeah, wow, those are terrific questions and outstanding questions. The original paper, we did PCR in those highly purified cytoplasmic fractions. And we actually had to use pretty short primers to get a good signal. And so we concluded at that point that it was fragments of mitochondrial DNA that we're getting out. And there's older papers on mitochondrial DNA getting out through the mitochondrial permeability transition that also claim that there were, 100 to 500 or something like that fragments that can escape mitochondria. But I just showed you the whole nucleoid coming out. So we haven't sorted that out yet. So and the nucleoids have TFAM on them. So I don't know if it's both. If there's nucleoids that are coming out in a compartment and maybe they're not accessible to PCR, or fragments that are coming out. And there's reports in the literature under different release mechanisms where it's fragments versus the whole thing. And I think we just haven't sorted that out yet. But I don't know the answer for TFAM yet, conclusively.
And then Jan asks, thanks for this talk. And does UL12.5 overexpression also induce C-gas sting signaling? And if so, is this a sustained response even though the mitochondrial DNA disappears over time?
No. So yes, it does. And once the DNA is gone, it goes away. So in fact, and we even showed that in the TFAM cells, if you deplete the mitochondrial DNA, you can reduce the signaling as well. So you do need the empty DNA to signal. And UL12 goes to the matrix. It actually binds to another nuclease. It looks like a nuclease, but it doesn't actually have nuclease activity. It has to bind to another nuclease to degrade the DNA. So I think in that case, that's how the DNA is getting degraded in that case to some degree. For TFAM alone, I don't know what's happening and how the DNA is getting, if it's getting degraded. I guess I just answered that. I don't really know if it's getting degraded or not. Trying to figure that out.
Two more quick questions and then we'll move on.
Okay.
And then UN asks, are there other physiological or pathological stimuli causing mitochondrial release, DNA release, in addition to DNA viral infection?
Yeah, actually, I'm going to show you that. So I'll show you several more examples, and we wrote a review on that in 2017. There's another recent review by Stephen Tate's group, many different conditions. So I'd encourage you to read those, and I'll cover them briefly going forward.
And Jonathan Friedman asks, do you have an idea of what the inner membrane is doing to allow the release of the mitochondrial DNA?
Well, the one release mechanism that has been reproduced by multiple groups now is that when you induce basically caspase independent cell death, if you enact that, you get a mitochondrial DNA driven interferon response. And in that case, what happens is you get macropores of back and backs on the outer membrane of mitochondria. And these things become huge. This is Stephen Tate's work and Ben Kyle's work. And the inner membrane actually herniates through those pores and gets like a balloon out into the cytoplasm. And then their contention is that that is susceptible to breakage, and that's how the DNA gets out. So there's an extreme case of the inner membrane, squeezing through a hole in the outer membrane and breaking in the cytoplasm. I don't see, the data for that are really good. I'm not saying it's wrong. It's definitely not wrong. I saw the movies, it's happening. But I'm curious as how more things don't get emptied into the cytoplasm in that case, right? You're bursting open the inner membrane. It seems like everything would go out, but that doesn't seem to happen. So I don't know. We haven't looked at the inner membrane too carefully yet in our cells, so I don't know the answer to that.
Thank you,
I will say,
Gerald.
okay, I'll probably move on from there. Should I go ahead and go?
Go ahead, thank you.
Okay. All right, well, this is sort of the second half of the story. And a lot of the work I'm going to talk about now is from Jun Wu, a previous Yale graduate student in my lab, who is now at UTSW and Ralph DeVeridinus' lab, and is probably on this call, I hope. So I'm going to tell you a little bit about his work. So he followed right after Philip, trained a little bit under Philip, and this is his story. So I just told you that release of mitochondrial DNA enacts an antiviral innate immune response. But we started looking into this deeper, and it's not exactly like an antiviral response. There's some key differences. And I'm going to show you what those are here. So first of all, even though we're getting upregulation of interferon-stimulated genes quite robustly, we were not getting upregulation of the interferon genes themselves, which you would expect. And we did all sorts of experiments blocking interferon with antibodies, interferon receptor with antibodies, conditioned media experiments, et cetera. And these cells are really not making any appreciable amounts of interferon where you could stimulate another cell. So that was kind of weird. We're getting half of this response. We're getting ISGs, but not type 1 interferons. We also didn't get activation of NF-kappa B. So here's some IL-6, IL-1 beta, TNF, are not upregulated in these cells, basically. And so the question is, the mitochondrial, unlike a viral, which should activate both of these pathways and give you a huge interferon response, where the mitochondrial response seems to only be giving us the ISGs and splitting this signal somehow. So we really wanted to look into that a little bit further. And so one prediction from that idea is that the signal should be independent of interferon and the JAK-STAT pathway. Because for normal interferon signaling, interferons are released, they come out, they bind the interferon receptor, you activate JAK-STAT, phosphorylate the STAT complexes, and then those are the transcription factors that drive the ISG response and other gene expression patterns downstream. And one of the key transcription factors in this pathway, this is the so-called ISGF3, which is a heterotrimer of STAT1, STAT2 phosphorylated, and IRF9. And so if you look at this diagram, John surmised that, hey, if we want to see if it's really interferon independent, the transcriptional response we were observing, let's knock out STAT1 because it's needed for all of it's needed for all the responses. And so he crossed the TFAM animals to the STAT1 knockouts. And the expectation was that it wouldn't have any effect, right? Because it's independent of interferon. And of course, this is why you do experiments, because you don't always get the result you expect. When we did that, in fact, they were indeed dependent on STAT1. So here's the normal response, STAT1 knockout. Some of them were still upregulated, and those are regulated by RF3. All right, so that's not what we expected. So how the heck are we getting interferon stimulated genes? Why is the STAT1 dependent, et cetera? So we delved into this a little bit more. And I convinced him not to quit his Ph.D. because he was quite dismantled at that point. But the bottom line is, so we looked for phosphorylation of STAT1 in these cells. And what you can see is that here's the wild type, here's the het. So you have more STAT1 in the het, but it's not phosphorylated at the canonical site by JAK, even though we could detect it if we added an agonist. So STAT's not phosphorylated. And so doing a little more digging, there's actually an unphosphorylated version of this ISGF3 factor that John Stark and others had identified. And so we decided to ask the question, is it this factor? And so it made sense. If you knock out STAT1, you don't have this anymore. And I just showed you those data. But if you knock out STAT2 and IRF9, same thing. You get rid of the response. And there's no additive effect of knocking those down with STAT1. So it appears to be this complex that's driving this ISG signature. Furthermore, Andy Min at UPenn and others have shown that there is a subset of interferon-stimulated genes that is associated with resistance to cancer chemotherapy, radiation, even some targeted and immunotherapy. So a weird it's not all the ISGs, a subset of ISGs that leads to this cancer treatment resistance phenotype. And they called it the ERDS subset, the interferon-related DNA damage resistance signature. Turns out that our subset overlaps quite a lot with the ERDS subset, and that subset also overlaps quite well with what Stark and others showed was the UISGF3 subset. So that subset has a unique ISRE that USISGF3 binds to. And so this is a unique subset of ISG. So we were always struck by that because there's hundreds of ISGs. We only got 40 or 50 upregulated in the TFAM. So it's this weird subset. So that made John really ask the question, well, if it is this subset, maybe these cells are resistant to DNA damaging agents. And so this is an experiment with the TFAM het cells that he did with doxorubicin, DNA damaging agent. And you can see quite clearly that they are more resistant to cell death from doxorubicin and apoptotic cell death from that. So that's cool. He also went on to show that this is due to enhanced nuclear DNA repair. So the mitochondrial status is affecting the nuclear DNA repair status. He did this work in collaboration with Peter Glazer's group at Yale. And what we're showing here are DNA damage foci in the nucleus now. So this is H2AX and P53BP1, which form foci when there's nuclear DNA damage. And you can see that if you add a DNA damaging agent and look at the kinetics of formation of these foci, that the TFAM cells form these quicker. And he did the opposite experiment of asking how fast they went away. and they go away quicker too. So essentially you're shifting the whole DNA damage response and repair kinetic to the left in the TFAM cells. So this led to getting back to why we have so many copies and why do we have a damage-prone mitochondrial DNA genome. And our last line in his Nature Metabolism paper is proposing that mitochondrial DNA is actually a genotoxic stress sentinel molecule. So the idea is here you have a genome that can sense damage before the nuclear DNA gets damaged and send signals to the nucleus to protect it. And why would you do this? And why do you have so many copies, and why does this work? It works because there's no way you're going to damage 1,000 molecules. And mitochondria, because they can divide and exchange genomes and get rid of bad mitochondria and the damaged genomes through mitophagy and autophagy, once the signal's gone, you can get rid of those genomes and replace them with the ones that are still there. So it's kind of a neat system that we're proposing, that mitochondrial DNA might be able to act as a stress sentinel. And when it does, the idea is to protect the nuclear genome. Why do you want to protect the nuclear genome? Because that's where all of the goodies come from, right? So you want the mitochondria are just as interested in maintaining a pristine nuclear genome as the rest of the cell. So the nuclear genome doesn't have that. It has to have all these repair pathways ready to go because there's no way to sort out the damage. You have to find it and repair it. So is there anything consistent with this idea? And so these are some experiments that John did in collaboration with Laura. And now we're just, again, looking at wild type MEFs treated with doxorubicin. And here you can see something that's going to look quite familiar is that you get enlarged nucleoids and elongated mitochondria when you treat with this DNA damaging agent. And you get release of mtDNA into the cytoplasm. Furthermore, you get upregulation of interferon stimulated genes. That's cGAS, STING, and TBK dependent, but not MABs dependent. Sound familiar? Furthermore, if you deplete the mitochondrial DNA, this is related to the question that was just asked, before you do that, you get a much less robust ISG response. It's not completely gone, but because there's probably nuclear DNA damage, it's activating cGAS. But a major portion of the damage is going away when you deplete mtDNA. And if you just take a version of doxorubicin that's targeted to mitochondria and only cause mitochondrial DNA damage to begin with, that induces an ISG response. So the mtDNA damage seems to be sufficient for this. Finally, John, you can read it in his paper, took TFAM knockdown melanoma tumors, implanted them in mice, and showed that in vivo they're resistant, partially resistant to doxorubicin. So this could be a mechanism of chemoresistance. And this is his paper. That's what I just said. And then I just wanted to add, we're writing up a little opinion piece on this right now, and I put together a table of some of these ISGs that are upregulated. And you can see that many of them have known or postulated roles in nuclear DNA repair. And so one of the ones we focused on were these PARP factors, PARP9, PARP14. There's another one called PARP12 that's upregulated doesn't have any known function in DNA repair. I don't know if that's right, actually. Anyway, he showed that the DNA damage response in his paper is partially dependent on PARP9. So I think collectively, a subset of these subset of ISGs are involved directly in nuclear DNA repair and damage resistance. So again, completely in line with this genotoxic stress sentinel thing. I don't know why this is in here twice. All right. So this really leads to, since that discovery, it's now been shown by multiple groups. There's a nice review from Stephen Tate's group this year, that mitochondrial DNA can activate not just the cGAS STING pathway, but other innate immune signaling pathways. In fact, it was known before that it can activate TLR9. It activates cGAS STING, as I just told you. It can also activate the inflammasomes, lead to IL-1 beta and IL-18 production. So this is a burgeoning area, many different circumstances where mtDNA is being released and different sensors are getting activated by that signal. And a huge area right now is also that there's circulating cell-free mitochondrial DNA, which activates TLR9 and can lead to systemic inflammation as well. And so, as I alluded to, we wrote a review on this a couple of years ago. Here's just a few, and this list just keeps growing. There's multiple physiological and pathological states where mitochondrial DNA-mediated inflammation has been implicated at this point. And interestingly, several viruses also lead to mtDNA release in addition to HSV. Zika, dengue, and even SARS-CoV-2 is a paper on bioRxiv showing that mtDNA is released in response to SARS-CoV-2. So this is a really cool area, a really fascinating area in terms of the role in pathology. So I want to end with some unpublished data, which gets at the question of what's the potential role of these pathways in aging. And here's a paper from 2014 looking at a human cohort of different ages. And what they demonstrated is that after the fifth decade of life, there's a trend for an increase in circulating mitochondrial DNA systemically. And that's correlated, those with the highest levels of circulating cell-free mtDNA had higher IL-6 and TNF cytokine production, pro-inflammatory. So that's kind of interesting. And they suggested there might even be a familial component to this. So we've been doing a few experiments to try to get at the aging aspect, and I'll just cover those real quickly and we'll be done. So the first thing we did is we took all the Sains, another Yale graduate student who moved to Salk with me, took tail fibroblasts from young and old mice and cultured them in vitro to do some cellular aging studies, senescence studies. And as you would predict, the older fibroblasts reach a proliferative plateau earlier than the younger ones. They show senescence accumulation earlier. And what do they show? They show increased interferon stimulated gene expression, but not an increase in interferon. So that sounds like the mtDNA signature to me, and we're trying to test that out. A similar example, if you just take human fibroblasts, dermal fibroblasts, and do the same experiment, just grow them, replicatively age them in vitro, and until they start to senesce, you get depletion of mitochondrial DNA in these cells, you get upregulation of ISGs, but no upregulation of the interferon genes. So again, sounds like an mtDNA signal. She took cells at about this point here at passage 18 and depleted the mitochondrial DNA in those. And you can start to see the ISGs come down in those cells. Not all of them, and I don't know why, but this is actually a pretty hard experiment to do. But again, this is very preliminary. We have a lot to do here, but I think it could be that the mtDNA release pathway is being unveiled during senescence. So what about aging? So, and is there a role for this pathway in aging? This is a question I'm really hot on the trail of right now. And the reason I'm interested in it is because, you know, one of the main players in aging is low-grade chronic inflammation, which, you know, impacts all of these age-related diseases, but nobody really knows what all the root causes of this chronic inflammation with aging is. And so we're postulating mtDNA as part of this issue. And of course, there is a mitochondrial theory of aging, and the mitochondrial theory of aging states that, you know, usually that mitochondrial reactive oxygen species damage the mitochondrial DNA, damage mitochondrial membranes, et cetera. That leads to dysfunction in the oxidative phosphorylation system and the electron transport system, leading to even more ROS production. And you sort of get this vicious cycle that leads to, over time, to cell tissue damage and death. But I think, you know, people argue about the validity of because ROS are also signaling molecules. Our own lab has shown that a little bit of ROS actually can extend lifespan in yeast, and others have shown it in worms and flies. So it's a complicated mix with the ROS, but what is clear and everybody agrees on is that mitochondria get dysfunctional with age and they get damaged with age. And if that's true, I think that mtDNA damage could lead to the release and pro-inflammatory aspects of aging. That's our hypothesis. I have not shown that at all. But the idea is, it's not just the DNA that's pro-inflammatory for mitochondria. Proudfoot and more recently Agnel Sphere just published in Nature that the mitochondrial RNA can also activate MDA5 and RIG-I, RNA sensors in the cytoplasm. Michael Caron and others has shown that oxidized mtDNA signals differently, activates the LRP3 inflammasome. And remember, mitochondria came from bacteria, so they have many so-called DAMPs, damage-associated molecular patterns, that activate, if they get out of context, can activate the immune system, the innate immune system, as if a bacterial infection. So this is something that we coined, Laura and I coined in a little commentary on Richard Ewell's paper on Parkinson's disease and the role of this pathway in Parkinson's disease. We called this mitoflammation. We got mitophagy, and we figured, what the heck, we'll go for mitoflammation. But I'm going to go one step further and end the talk, and that is, what about mitoinflammaging? So the idea that inflammation is involved has been called inflammaging. I want to call it mitoinflammaging. And so here's some ideas on where that chronic inflammation is leading to aging theory comes from. It comes from infections, it comes from increased DAMP production, like I just proposed. Senescent cells secrete cytokines and other pro-inflammatory molecules. This is where it's thought to happen. But if you really think about it, I think there's a major role for mitochondria in all of these. So I've already shown you that mtDNA and other DAMPs can activate the innate immune system and inflammatory pathology. Mitochondrial dysfunction can activate its own version of the SASP that leads to a particular, Judy Campisi has shown, this leads to a particular milieu of cytokines that are released. I've showed you, and many have showed subsequently, that pathogens can lead to mtDNA release. Immunosenescence, mitochondrial dysfunction wears T cells out, causes dysfunction, and leads to a state called exhaustion in T cells, which limits their function. Obesity is definitely a condition that leads to mtDNA release. And I don't know about the gut microbes, but I can only imagine there's communication between the microbiome and mitochondria in this regard. And then lastly, if the DNA can get into circulation, you can really see how you can get systemic activation of these pathways that could contribute to aging. So the last little teaser piece of data I have. So we for a long time, have been aging TFAM mice. And two things. In vivo, we could see it in the MEFs and in BMDMs, you could see this ISG signature, but if you do like liver or other tissues, you don't really see a robust ISG signature. And we could never quite figure that out. And we aged them, we did a couple of aging experiments a while back, and we still didn't really see anything. But now we've aged them to where they're really old. So these guys are 20 months old. And now you can see this ISG signature start to pop up in these mice. And this is super preliminary. This is our first takedown of a small cohort of mice. And these are all male. But what we're seeing is what are cancer phenotypes in the liver of these mice. So we're seeing inflammation, we're seeing lesions. I think this is a tumor, we're sectioning it right now. We get increased Ki67 staining compared to the wild type. So it looks like this ISG signature becomes more robust with age and in the TFAMs, and it may promote a cancer phenotype. So I'll leave you with that and stop there and take more questions, but I just want to acknowledge my funding sources, which are shown here. I think I acknowledged most of the people for their work along the way, but this couldn't have been done without some really amazing collaborators. My wife, Sue Keck, is a longstanding collaborator, helped me a lot with a lot of the immunology and viral work. She's been part of the story the whole time. Peter Glazer's group at Yale was instrumental with Sebastian Nowak doing the cancer studies in the TFAM melanoma cells. And I won't list them all, but the rest of them are there. And the collaborations have been really great, and we're continuing those since we moved to Salk. So I'll stop there and be happy to take questions if there's time. Where are we at? Just about, yeah.
Okay, we have a few questions. UN asks, is it possible for mitochondrial DNA to be released to the extracellular space and sensed by other cells? So I think you kind of talked about this, but perhaps you could comment on that.
Yeah, no, definitely. It's not clear to me how it's getting out, all the way out, but it does. And it's also gaining access to the circulation. So it's getting out and then getting into circulation somehow. And I think the pathways for that are just really not understood yet, but it's definitely, there's a really nice paper on lupus where they show that the DNA is getting out and activating dendritic cells, for example.
And John Abrams asks, do you think there are specific tissues or cells that are the source of the cell-free mitochondrial DNA?
Yeah, well, I do, but I don't know why. I don't know what the basis of me thinking that is, but we are collaborating with a physician at UCSD named Mark Hepikowski, and he has shown, he thinks the kidney is a particularly relevant source of the circulating mitochondrial DNA. And in some acute kidney injury models, he sees increased mtDNA in the cytoplasm. Brett Kaufman and his colleagues just published a paper knocking TFAM out in the kidney and showed some of these things are happening. So for some reason, the kidneys are packed with, the tubules are packed with mitochondria. They're kind of prone to hypoxia and other stress. And so I think that's one cell type that I think could be a key contributor. And the reason he came to me is he works in the ICU. He's an ICU doctor. And in the ICU, kidney dysfunction often drives systemic inflammation that leads to death in the ICU, according to him. And so he really thinks that the kidney is sort of is one of the main instigators of systemic inflammation from mtDNA. He hasn't proven that, and we haven't proven that, but that would be one guess. I think neutrophils, because they can release the nets, the mitochondrial DNA as part of that pathway, netosis pathway. I think so neutrophils and maybe other immune cells might be another pretty big source of that. But interesting, red blood cells can actually seem to soak up the circulating mtDNA and prevent signaling. There was an interesting paper about that. So it's a great question, John. And it was John, right? John.
Yes.
And a lot of work to do there, I think, still.
Okay. And Beverly asks, are there mitochondrial DNA haplogroups? Are certain mitochondrial DNA haplogroups more pro-inflammatory than others? And does the mitochondrial uncoupling reduce or increase release of mitochondrial DNA?
Well, thank you for asking the haplotype question. This is like one of my biggest peeves with geneticists. We do all these GWAS studies and they never look at the mitochondrial haplotypes to see if I shouldn't say never, that's an overstatement. They're largely ignored in terms of driving pathology or even other physiological responses. We wrote a little section on that in our review article about the haplotypes. So there's a few really, there's nothing I know of a particular haplotype being more inflammatory, but it would be interesting. It's an interesting idea, and I would like to delve into that. But thanks for that question. And I don't know the answer to the uncoupling question. John, are you on? Do you know? He might have done it. No, he's not there. He's not going to rescue me. I don't know. I mean, uncoupling, if it's severe, will lead to mitophagy. And there is some indication that maybe even when mitochondria get into the lysosome, that the DNA can get out from there too. So, but I don't know the answer to the uncoupling question. Yeah, I'll have to look into that.
Okay. And then in the het mice, do you see distorted interference between the ER and mitochondria and deregulated UPR responses?
Do we see distorted? communication between ER and mitochondria?
Interference, yeah,
Oh,
between the ER and the mitochondria.
well, you're trying to get me to divulge data I don't want to divulge. But so it's a really great question. You know, Jody Nunari showed that, and Gia Volz showed that the ER marks sites of mitochondrial fission and is actually physically involved in mitochondrial fission. Also, it marks sites where mitochondrial DNA replication is going to start, according to their model, Jody's model. So I think this is part of the reason I have this idea that the TFAM cells have, what I think their model is that you start replication, you mark the sites with the ER, you start replication of the genomes. Then when they're done and segregate, the ER enacts its fission role and splits the mitochondria right at that point so that you inherit the genomes, just like a bacteria would. You want to make sure you inherit your genomes, right? Or any cell would. So I think that's what's going on. And so we are very interested. I think the reason that we have this hyperfused phenotype is because they're not dividing, they're waiting for something and they're not dividing. And we do see disrupted mito-ER interactions. We also see really a lot of ER, once the DNA gets out, the ER seems to be right there around it. So there's a weird membrane dynamic thing going on there when the DNA gets released, and it does involve the ER. And we're still sorting that out. We're, that's going to hopefully be our next paper on this, is to try to fully understand the nexus of the ER and the mitochondria and that release mechanism. What I can tell you is that in our hands, the release is not the back-backs activation. And more recently, a paper came out showing that VDAC pores can also lead to release of mitochondrial DNA. We don't see a role for VDAC either. So the TFAM release mechanism is something it's been a hard nut to crack, but it does seem to involve disruption of the ER communication. And we're almost there, but not quite. Great question.
To that effect, though, is there anything specific about TFAM or other mitochondrial DNA binding proteins also give you similar phenotypes where you have a knockout? Do they give similar phenotypes as the TFAM?
Yeah. Some of the nucleases, Endo-G, can give you this phenotype. And that's how the VDAC paper actually showed. In fact, it's kind of funny in that paper, they say, well, the TFAM has less DNA, so it's not really clear why it's relevant. And then and the knockout is just confounding for some reason. And they use a knockout of Endo-G as if that's different. So they knock out Endo-G and get released to the VDAC pores. So they're getting it released through a different mechanism than us, and I don't understand that. But we've knocked out a few. I think Twinkle, which is a DNA helicase, does it. And I've seen people at meetings suggest that mutations in Paul gamma also, which is the main mitochondrial DNA polymerase, can also, under some circumstances, lead to mtDNA release. And Philip West just published a paper on the, who was at Texas A&M, by the way, as an assistant professor, has a nice paper on the Paul gamma mutator mice that make more mutations in the mitochondrial genome and have a premature aging phenotype, and shows that type 1 interferon signaling is important for that phenotype as well. So yeah, I think there's multiple disruptions in the mtDNA machinery that can lead to this.
Okay, great. Well, thank you, Jerry, for that wonderful talk. We had a lot of questions and a great discussion.
No, that was really fun. Really, yeah, almost feels like a real meeting with that many questions.
Well, thank you. I believe you have meetings